basically, it's going to be a little demo, which is improvisation of small RNA isolation, because we isolate from different type of tissues where small RNA is less or more abundant. And today, since we cannot work with radioactivity here or something, we're going to isolate the most abundant class of small RNA, which is RNAs, pakitin RNAs in mouse spermatogenesis. So you can visualize them on a gel using cyber gold. So, and it's you're just gonna get a little bit taste how we do it, and yeah. Meanwhile, actually, gel is running. We will discuss. I mean, I will give you a little bit intro about small RNA silencing pathways, and we'll give you like theoretical aspects of small RNA cloning, making libraries, and at the end I will share a protocol which we have, and we can basically discuss details. For example, if you want to barcode your libraries and do other things. But realistically, it needs 3 days to really make a library. So, and I have only an hour to.
They've suffered through 2 days of library prep.
Yeah, and I mean, and this one is the longest one. So RNA-seq, it's a simple one. So or genomic. All right, so.
Do we have a protocol posted on the wiki?
Yes, we're going to get protocols.
I have a protocol. Yeah, you will get a copy of protocol. You will get a copy of this presentation and an article, original article about pakitin RNAs. We'll kind of try to reproduce picture which they got there. So I never done it before. So no, but I do have some backup lanes. Yeah.
Okay.
All right. So what we'll do like canonical type of small RNA isolation using polyacrylamide gel. It's so far the best way of isolating small RNAs, because you're really precise in the size small RNAs which you want, and small RNAs which we have. I mean, we are working on small RNAs which are associated with Argonaute proteins. Their size can vary from 90 nucleotides to 35. So basically, we want to be in this size frame. Of course, there are some other ways of small RNA isolation exist, like size columns, but they give you a range from, let's say, 5 nucleotides to 200, and you have majority of degradation products from the most abundant RNAs, like sTRNAs or ribosomal RNAs. So I do like this way better. There are Alexa kits exist. They give you some small RNA libraries, but level of the contamination by ribosomal tRNA is much higher, and usually if you really want to be deep or you want to barcode your libraries like on one Alexa lane and let's say put 10 libraries, you really want to have a good quality of RNA, so that's the way to go. And now what I will do, I will have size markers, and I will have 293 cells which don't have any RNAs. It's going to be mouse testicles, and after that.
Yeah, we can't hear you.
Okay, I'm sorry.
Really talk loud.
Yeah, so basically we'll run 2 real total RNA samples. So one sample is going to be 293 hex cells which do not have pakitin RNAs, and another sample is mouse testicles, like total mouse testicle RNA. So
when you did total RNA, how did you purify the total RNA?
Yeah, it's trizol. There is also Nirvana kit exists from Ambion. So yield from Nirvana kit usually beta.
What's the name of that one?
Nirvana, M-I-R. Yeah.
Okay, not marijuana.
Well, everyone, yeah. Well, everyone want to hear what, yeah. No. Yeah.
I noticed that his RNAs are at room temperature.
Yeah, RNA is at room temperature because I want to tell you that I already precipitate RNA, measure RNA concentration. It was in water. Now it's a mixture with RNA loading dye. RNA loading dye contain formamide, so formamide stabilize RNA. And before loading on polyacrylamide gel, usually you heat RNA at 90 degrees for 2 minutes just to denature secondary structure. It doesn't mean that you do not have any secondary structure even in 7 molar urea gel. So Yeah.
So is it a standard like formaldehyde fluoramide gel?
No. It's a polyacrylamide gel. It gives much better resolution. Basically, it gives you one nucleotide resolution. That's what you need when you're really playing with these sizes. So.
Do you have formamide in the gel?
No, there is no for urea and yeah.
What percent gel?
This 15% gel just give us a little bit time to talk, so you can do 12% gel, but yeah.
Everything will be in the protocol for sure.
Okay.
Yeah.
No, just help us
Yeah,
understand.
No, I mean I can actually pass around. I have
We'll get all of that
yeah
written down.
yeah.
That's great. So the plan is to load the gel and start it running, right?
Yeah, the plan to load the gel started running. While gel is running, I'll give you a little bit intro about small RNAs, and after that, yeah, we don't have here the board. I could write exact steps of small RNA clonings, but probably I will. Yeah, you will have to listen to me. You don't have any whiteboard here.
Yeah,
Oh,
we
you.
have
Oh,
one.
great.
That we have.
Then actually solves all our problems. So yeah, I will load the gel. So basically, this samples already were denatured and ready to load. So basically, yeah, I
Do
will.
you purchase the gel or do you pour it?
I mean, if you want, you can purchase, but
Okay, you just, it's not
Yeah.
a gradient gel or anything.
no, it's not a
So
gradient
you
gel.
just pour it yourself.
There is no purpose to do it. It's like pretty much is like old school sequencing gels. So usually use 5% or like, but here's a 15%. So basically,
But it's super thin.
yeah. No, well, actually, in fact, actually, gel for small RNAs are not that thin. So there is 0.75 millimeter spacers.
Oh, okay.
Because I mean, in fact, I mean, you can clone small RNAs from the RNA, total RNA probably from 500 nanograms to 20 micrograms. 20 micrograms is ideal concentration of total RNA, amount total RNA which you put on a gel. But I mean, a lot of people working with tiny materials and a couple cells. It's possible to clone small RNAs from 200 cells, but yeah, it's
Question. So if you're trying to resolve 21 mer versus 22 mers, is that a big enough gel?
yeah, it's a big.
It is?
Yeah, I mean, if you like try to stop gel around 5, you'll see, because what I have here, I have a mixture markers 19, 24, 28, 30. You will see the difference between 28 and 30. So in
What
fact,
size are you? Because you said they're the kiwi RNAs, or which size are the ones that you're?
the size which we'll deal with, it's gonna be from 25 to 30, 32, something like this. Yeah, so and basically I have a lot of small RNA markers. You'll see it on the gel. Any other questions before? And I mean, now it's going to be simple loading, so it's. I can mumble something when I will load, but it. Oh yeah, actually this TB, 1x TB, and yeah, samples. And samples and format.
Yeah, if you can't see that too, but that's kind of light. Yeah. No, but I can't see it, so I think it's just loading a gel, right? It's okay.
Yeah, I mean, I'll try to see it in a way that you can see.
It's okay.
And it's a handmade gel. It's not that perfect. 'Cause,
Loading a gel. So the thing I would be curious about, like, do you have to like worry about RNA-free polyacrylamide, RNA-free TBE?
no, we do not care, and I don't know.
So,
Yeah,
Rita? Cool.
Kind of even RNAs.
Okay.
I mean, only one thing which you need to take into consideration is having gloves when you're working with RNA. And I don't know, with me, I probably don't have any RNAs on my hands. I didn't really have problems with RNA degradation in like last 10 years. Probably that's.
Yeah. So it's good. Yeah. But it also might be easier if you isolate small RNA, right? You get some degradation.
Oh yeah, they're already like getting smaller.
Well, that's tricky.
That's probably I moved.
Honestly,
Yeah, that's probably I moved to smaller RNA field because.
No, it's true.
it is true.
How do you separate
The way how you separate degradation products in general, but of course there are exceptions. So basically, usually degradation happens sort of randomly, and what you get in 3' phosphate, usually that's and 5' hydroxy group, and yeah, degradation products. Of course, there is some RNAs which have 5' phosphate and 3' hydroxy group, but small RNAs which are functional, they do have always 5' phosphate and 3' hydroxy group. That's how actually, basically that's, and we use these properties to clone small RNAs. So, and we get not that much degradation products, just because we use these chemical properties of small RNAs.
Okay, and those during the library prep, they won't get adapters.
Yeah, during the library step, getting adapters, and I will explain later how it works. So, and yeah, I mean, it's, yeah, it's basically everything based on
Just small RNAs get converted to DNA first.
no. First you add 5' and 3' adapters, and I mean using RNA ligation, and after that you do RT, but
See my scheme? I know you can do RNA.
yeah, a lot of viruses doing this all the time.
You're not the first person to try it.
No, I think first person which tried it was like Ph.D. or something like this, and
So the only other thing is we'll have to take a walk over to the UV box in the
yeah.
to see the gel. We didn't bring it over, so.
I mean, theoretically, I can try to bring it over meanwhile gel is running, but
Yeah.
whatever way it works.
So I'm assuming you boil them
Ye-
a while ago. You don't worry about leaving them on the bench.
No, in formamide usually RNA is pretty stable. So, and actually I did, and yeah, I'm saying that RNA is formamide is pretty stable, and I did boil them probably 20 minutes ago. So,
No, I'm more worried about reforming secondary structure despite.
yeah, the small RNAs for sure, it's very difficult for them to fold into secondary structure because I mean they're small, right? For tRNAs, they will have secondary structure no matter what. So it's like this gel even 7 molar is
7 molar urea?
yeah.
Oh, that's amazing.
Because
So
what we used to do
tRNA is
is
still
boil
functional.
them and
And
then
put in
immediately put them
ice.
on ice.
Yeah.
To kind of try to reduce the.
Yeah, you're reducing, but you're not completely killing
Oh,
it.
you
So
will
yeah,
win.
I mean RNA likes to fold into.
Okay.
Yeah, and
You started the run already?
not yet. So basically, I mean I have to put top chamber,
Yeah.
and I hope it won't leak. But I pre-run gel, so it didn't leak. So you know what I forgot, and I will have to go to McClintock. I forgot to clicks to close.
I know what it looks like.
I mean they just look like this. So
Where will they be, on the ground floor?
they're gonna be on the ground floor in a hot room.
In the
So
hot room.
yeah,
Oh,
you can ask.
wonderful.
Yeah,
Love the hot room.
yeah,
All right, I'll try.
yeah. All right. Yeah. Meanwhile, we're waiting for more. I could bring them. Yeah, we may start presentations. So.
Mine I can just switch, because all you're waiting for is a piece of equipment to
Yeah,
hook
yeah.
up and then you just hit
Yep.
go.
Yep.
So is it running now?
No, because actually I need to finish assembling like top chamber, and I forgot like 2 little screws to make it seal.
Mine is just.
Oh yeah, so you can't put the buffer right now.
Well, I can pull, but we will not be happy with
You don't
results. So,
want to flood.
all right. And we don't have pointer, right? That's okay. All right. So. Oh. Yeah, it's gonna, if you can. Or I can stay on this side. I'm just probably right, so that's why. All right, so it's fine.
Okay.
Yeah, it's gonna be a little bit change for my brain, but so 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 I mean, since we don't have a lot of time, I'll just give you a little bit intro on the small RNA silencing pathways, actually in plural, and why they exist. And first of my slide, basically what I want to stress out, that our genomes are full of transposable elements, and in human we have more than 50% of repeats, which considered to be junk RNA. And here just examples of different transposable elements, and this word cloud. So basically the bigger word, the more representation, for example, for mouse L1MD transposable element. So, but I mean, basically we're full of transposable elements, and we have different protection mechanism against these bacteria develop CRISPRs, which are RNA-based immune defense against them. There is also DNA elimination, which exists in tetrahymena, which also kills all repetitive sequences and eliminates them from the genome. There is also heterochromatin silencing, and the another mechanism which I will talk today, it's a small RNA silencing through Argonaute proteins. And what Argonaute proteins are? So this crystal structure of Argonaute protein, that's model of the crystal structure of Argonaute protein. So here is yellow is a small RNA, red is the mRNA, and the rest of the scheme is domains of Argonaute protein. Basically, Argonaute proteins binds with the small RNAs, and based on complementarity of this small RNAs with mRNA, find mRNA and induce a cleavage of mRNA and eventually degradation. And idea is basically if you're not complementary to mRNA, you do not degrade this mRNA. So basically, small RNAs works as adapters which help Argonaute proteins to distinguish between self and non-self mRNAs in cell. All right, so yeah, I just shortly start. And
Sorry. I don't want to break it. So
oh yeah, sure.
I don't know how to put them in.
I mean, they work like screws, but yeah, I do it. Oops. All right, so once we start the gel, I'll continue. But I mean, basically, main point of small RNA is that the adapters which actually tell Argonaute protein what RNA it can degrade and what RNA it can't. All right, so I will fill buffer in a top chamber. So, and we pretty much ready to go. We need to connect chambers. So, and the way how we run gel, so basically gel has to be a little bit warm. So if it's warm, it's denature RNA more. As I mentioned, you cannot denature all RNAs. But it has to be warm, so, and, okay, I can run. So plus, minus, everything's correct. All right, and meanwhile gel is running. Just let me make sure that it will run.
What voltage do you use?
Actually, I'm setting up based on watts. So, but voltage is about like 900 to 1,000 volts. Yeah, but everything based on watts. So, and I'm making sure that gel is not going hotter than 60 Celsius degree, because usually glass breaks after. So, yeah.
Plus multiple plates too.
Yeah, I mean, one secret, you can put a metal plate, right, which goes up.
That uniform.
Yeah, no, I mean, all
Or
techniques.
you can have like recirculation of
Or you can, yeah, I mean, there are all techniques exist which actually, yeah. we are not using anymore.
Hey, come on, they were not that bad.
No, I'm saying they're great, so it's like.
I have a question. I know there's these things called Kiwi RNAs.
I will go to this point. So basically, this is actually a description. Since you asked this question, so this is actually domains of Argonaute protein, and Argonaute protein has a pass domain which binds with 3' end of small RNA, and it recognizes 2', 3' hydroxy group, which actually indication that it's not degradation product. And there is another domain which is called mid domain, which binds with 5' phosphate group.
Okay.
Basically, Argonaute protein is making sure that it's not binding with random degradation products in the cell. And it also has a PV domain, and that's how originally PV proteins get this name, because it was found as conserved domain among all eukaryotic multicellular organisms, and no one knew exactly what it does. So, but what PV domain does, it's basically it's endonuclease, which makes a cleavage of mRNA at position 10 of the small RNA mRNA hybrid.
Can I ask another quick question about Argonautes? I know that they're usually fairly diverse. You know, they're like a big family.
Yeah.
Do they all do essentially the same thing? Or
No, and I will go to that.
You're gonna go into
Yeah.
okay, great.
So as you mentioned, it's a big family, and this is Argonautes, and typically in multicellular organisms, they divide on two subclades. One subclade called PV subclade, which predominantly expressed in germline, and another subclade is the ego subclade. And C. elegans develop specific C. elegans subclade, which has very weird biochemical function, and I will not go there. And there are also outlets, but yeah, and there is also subclade of prokaryotic Argonaute proteins, and recently it was shown that they required, basically they protect bacteria from efficient transformation, so with plasmid. So basically it's like still like genome defense against foreign DNA. So, and I mean, it was paper last year, but I mean, it's, I believe there are more, there is more research.
I'm not sure if you know the answer to this question. It's okay,
Yeah.
but I know that small RNAs sometimes cause like microRNAs cause translational
Yeah.
inhibition. Is that like independent of Argonautes?
No, it's everything is dependent of Argonautes, because what I tried to stress out that small RNAs are just adapters, which actually based on complementarity between, or based on whether they form duplexes or not, tell Argonaute protein whether actually it's your target or it's basically if you don't bind. Yeah, because it's basically it's kind of adapters which actually helps Argonaute proteins to find the right target. So.
Okay, so some Argonautes are translational inhibitors.
Argonautes actually bring machinery which can cause translational suppression. Even for some microRNAs, they can induce rather RNA degradation than translational suppression. And I mean, there is a bunch of combination, and please do not consider that Argonautes only one which is responsible for translation suppression. In fact, actually, it's pretty much like, I don't know, let's say it's kind of, basically when Argonaute find an mRNA, right, there is like kind of crosstalk. For example, majority microRNAs binds with 3' UTRs, and there is crosstalk with other 3' UTR binding proteins, whether you can actually degrade this mRNA or not degrade, or whether you can translationally suppress, and so on. It's not like only Argonaute which actually makes decision, but Argonaute designed to make a decision to do suppression, but it's kind of like a parliament which eventually votes for. Since actually I started, I'm actually trained as Drosophila geneticist, so I will give you introduction of two Argonaute clades based on Drosophila. And as I said, there is Argonaute clade, and Argonaute clade responsible for microRNA silencing, and here I'll give you an example of microRNA processing. Originally, microRNA is processed from the short double-stranded RNA hairpins, which are approximately 70-80 nucleotides, by enzyme which is RNase 3 enzyme, a dicer, into small RNAs, and these small RNAs find the target, and usually they partially complementary to the target, and they do have mismatches in position which actually usually are required for the cleavage. So that's why usually they do not cleave. But what they do, they use translational repression, and sometimes they attract other. Oh, yeah, that's great.
You have to talk louder.
Okay, and sometimes they attract other proteins which induce mRNA degradation. And basically, in flies we have Argonaute 1, which is responsible for microRNA silencing, and here Drosophila embryo, and RNA in situ hybridization shows that Argonaute 1 ubiquitously expressed through embryo development. There is another small RNA pathway, it's siRNA pathway, but still effector complex is Argonaute protein, and in case of Drosophila it's EGO2. And here we have long double-stranded RNA, which can be like 100 or like up to 600 nucleotides or even longer stretch of double-stranded RNA, which processed by dicer2, which also RNase 3 enzyme. And this Argonaute finds the target, and if it's complementary to the target, it's causes the cleavage. And again, it's ubiquitously expressed. And finally, pRNA pathway. Precursors for pRNAs is usually considered to be long single-stranded RNA, which process, we still don't know what process this long precursor, but finally it's incorporated into PV complex, and this RNA slightly longer than siRNAs or microRNAs, few nucleotides longer, and majority of them complementary to transposable elements, and they either induce transposable mRNA cleavage, or in some cases they induce transcriptional suppression. And here's the example of in situ RNA hybridization for the PV protein. It doesn't express ubiquitously through somatic tissue, but it's expressed in forming gonads, which finally will form either testes or ovaries.
Question, so the pi RNAs, are they processed by like a dicer-like activity? They have the same?
Most likely not, because dicer is double-stranded RNase nuclease 3 endonuclease.
So do those pi RNAs have distinct biochemical ends then, or do they have the 3'
They do have, I mean, in fact, actually, they do have, but they do have distinct end, but it's nothing to do with their biogenesis. Okay. So even if you remove enzyme which actually does post-processing modification, they still process, but they just do not have this modification. So
Do they have a 3' hydroxyl?
they do have 3' hydroxyl. You still can clone them using cloning, which we'll do today. So is it running? Okay.
So they don't have a double-stranded intermediate at any
For now, we consider not, because no one could find any double-stranded intermediate. So, and the pRNA pathway is a little bit more complicated, and I do not have time to go through, because I mean it has some build-ups which actually called self-amplification loop, which has a little bit double-stranded intermediates there, but yeah, for now on, it's like it's basically precursor single-stranded. No one could find any secondary structure. It doesn't form any hairpins, and there is still like $1 million question: who makes
So
actually
the
pRNAs?
precursor doesn't form secondary structure?
It doesn't form. I mean, you can find secondary structure if you use M-fold, but it's basically any secondary structure you shall find, they do not correlate with the regions where pRNAs are coming from. So basically, you cannot really find a pattern of pRNA production based on any predicted secondary structures there. Anyhow, so they express in a germline. And as Mona mentioned, now I'm working on a mouse spermatogenesis, and probably shortly I should give you introduction of this basically how it works. So in embryo you have prospermatogonia, which actually are just couple days before the birth, and these cells do not divide. They do not have any DNA methylation marks, but what happened in this couple days before the birth, you do get DNA methylation, and majority sequences which are methylated is transposable elements, just to make sure that they will not be active and will not jump to next generation. And after that, after birth, you have spermatogonia, which methodically divide, and eventually they go into meiosis. And at the end of meiosis, you have round spermatids, which already haploid cells. So basically, you have all these recombination events during Pachytene stage. And round spermatids eventually just change in shape, go like into elongated spermatid, and finally form mature sperm. So all process takes approximately 30 days. And why we are interested in this? So because we have, and this actually specific for male, for mammalian males. So we have 3 PV proteins which express differentially time-wise. There is MIV2 and MILI. In mouse they called MIVI because it's mouse homolog of PV. So basically everything comes with the M, but they still sounds like PV. So basically MIV2 and MILI, they express at early stages, and they're responsible for DNA methylation of transposable elements. And if you knock out MIV2 or MILI, what you have RS spermatogenesis just before meiosis starts, before Pachytene stage. And there is another protein which popping up at Pachytene stage, which called MIVI. And if you knock out MIVI protein, spermatogenesis just arrest after round spermatid, so you never go through to elongated spermatid, so of course mature sperm. And you have 2 populations of pyrranase. One is transposable element pyrranase, which is basically lines and signs, and it's predominantly expressed in embryonic testes and responsible for DNA methylation. And after that, this population sort of disappears, but another population appears, which is Pachytene pyrranase. This pyrranase, they coming from repeat poor regions, so it's pretty much intergenic spacers. And this population extremely abundant, and hopefully we'll see it today on a gel using cyber gold staining. So it's based on, to my knowledge, it's only one small RNAs which you can see by cyber gold without any radioactive labeling. And what these pyrranase do, we do not know for sure, but they have very interesting features. So they are not conserved in sequence, so it's very difficult to find any pyrranase which are the same in sequence between even mouse and rat. But what happened is that they're coming from the same synthetic regions. So basically, if you have a pyrranase clusters, and usually they come in as a clusters, and this is an example of the cluster. So basically, you have promoter, bidirectional promoter, which either transcribed in this direction or in this direction. And what you have here, you have a small RNAs, basically plotted here, and this is the count number. So basically, this small RNAs, it's one of our small RNA libraries. So this small RNAs will clone more than 12,000 times. So for example, this peak was cloned more than 9,000 times. But yes, you can see that actually this cluster is transcribed bidirectionally, so it doesn't have any genes here. Usually, genes somewhere around. And if you like go to the rat and try to see whether rat produce pyrranase from the same intergenic spacers, you can see the same. So for example, if you have here gene A and here is gene B, and you go to rat and try to find gene A and gene B, so you will for sure find pyrranase clusters which is coming from them. Even this sequence completely like diverse from mouse. So basically, almost there is no nucleotide identity between rat and mouse. Or let's say if you go to human, again, there is no conservation in nucleotide identity, but there is always conservation where in position of this cluster in genome. So,
Are they retained in mature spermatocytes?
sorry?
Are they retained in the mature spermatocytes?
No, it's in mature spermatocytes. Basically, maybe I should jump to this slide. So basically, at round spermatids, what you have a MIB protein, which actually localized next to the nucleus, and it forms so-called chromatoid body. It's a giant RNP structure with bunch of RNA binding proteins, and MIB starts degrading from round spermatids to elongated spermatids, and it disappears. And without MIB, this RNA is not stable. So, and you can see some remnants of this RNA, but it's most likely degradation.
Is it the same for microRNAs?
MicroRNAs, I saw a couple papers where they claim that they have microRNAs in the sperm, but seems like it's really poor if you compare it with oocyte is full of microRNAs. But I saw a couple articles where they trying to find microRNAs in mature sperm and trying to say this microRNA is required for future embryo development after fertilization, but I was not convinced with.
Some people say that about the messenger RNA as well.
I
don't know.
I mean, if you ask in my opinion, I'm not convinced, but these papers went through review. I mean, basically they went through reviewers were convinced. So, but I'm not really convinced about this because I'm coming from, I'm geneticist, so basically if they will show that this mutation like cause disappearance of this tiny fraction of microRNAs which left in mature sperm, and it caused like really dramatic effect in embryo development, I will believe. But I mean, so far it's, I mean, it's very difficult to do genetics in mouse and much easier in flies. So, but flies researchers, they didn't jump to this question yet. But
could it be involved? I mean, if it survives, could it be involved in transposon silencing in oocyte?
All right. So basically, the general answer for all non-mammalian organisms, yes. So basically, you need RNA pathway to silence transposons in ovaries, but in mammals, none of these mutants, either single or combination of them, do not cause. Or what I didn't mention is, basically, when you mutate PV proteins, you cause sterility. So basically, because of transposon element jumping, so you have a lot of double-stranded DNA breaks, and you do not progress to the end to mature oocyte or to mature sperm. And if you do the same with mouse and check females, females perfectly fertile, even males like 100% sterile. And in mouse, actually, in fact, Argonaute or clade is taking care of transposable elements. If you mutate Argonaute 2 in mouse, basically you see transposon upregulation in oocyte and basically they're sterile. But yeah, so basically RNAs are not essential for fertility in mammalian females. So, and today what we're gonna do, we will, this is actually example of polyacrylamide gel, which stained with cyber gold. And this one of the 3 original papers which were published on Pakitin RNAs. And here's, I believe, different type of cells which were sorted from testes. It's spermatogenesis cells, PSC, Pakitin spermatocytes, round spermatids, elongated spermatids, and LSP, I don't remember this abbreviation. But anyhow, so basically what we would expect to see today on a gel, and I hope we'll see. So basically it's going to be bent, which here's, you can see 2 RNA markers. they didn't have back on this time marker which is 30 nucleotides, and this RNA is a little bit above 30, but you can see them on a gel, and for sure we'll see tRNAs and some of these products probably ribosomal RNAs like 5.8 sRNA probably somewhere here. But yeah, basically for sure we'll see this part, and I hope we'll see this part. So yeah, that was actually a small intro. If you want to ask more about small RNAs, I can answer the questions. If you don't, I can go ahead and tell you theoretically how small RNA cloning works. And at the end of our class, I'll give Mona a USB with the small RNA cloning protocol which we're using in the lab, this presentation, and this original article about pachytene RNAs. So.
I just have one quick question. Like the precursors for the Kiwi RNAs,
Yeah.
are they all different sizes, or are they?
They're different sizes. So some of the precursors pretty long, 100 kb. So transcribe.
100 kb?
Yeah. Some of them splice, some of them non-splice. So basically, I guess based on my answer, you understand that we do not know a lot about rules how these precursors appear. And some actually of the precursors, in fact, 3' UTRs of the coding genes. So, and it's not clear why RNA is derived from these 3' UTRs. Yeah, but in general, general belief that they are single-stranded because we do not need dicer for their production. And as I said, even if you try to put in M-fold any possible secondary structure, there is no correlation between existence of secondary structure and RNA production from this region.
Right, so they're really different than a microRNA.
No, that's for sure. I mean, they do. No, they're different in terms of genetics. They need absolutely different genetic setup besides Argonaute protein. Even this Argonaute protein evolve specifically bind with this set of RNAs, and this RNA seems like appears when germline appear, because if you go to Saccharomyces pombe, it has Argonaute, but it's pretty much happy with.
So these do they bind to the Kiwi, like that Kiwi protein that you drew as part of the Argonaute complex?
Yes, there's a PV binds them, so it, as I said, it's pretty much as Argonaute protein, so it has also PV and PAS domain, so it's recognized 5' phosphate and 3' hydroxy group. So, and I mean, in general, Argonaute proteins designed smart to not target their own genome, because, I mean, if you bind with the same size of but degradation products, you can really cause a lot of, yeah.
Do you know, are these secreted at all?
Are they secreted? All right, I saw one article. It was published, if you're asking about small RNAs. So it was published in a journal called Science a couple years ago. And they were claiming that they could see secreted microRNAs in breast milk. That and they were kind of trying to drag this to the, even they couldn't see Argonaute protein, which actually made me a little bit suspicious, because in general, small RNA is not that stable. I mean, they're stable in formamide, but yeah, they, yeah.
I've seen something about microRNAs being exosomes.
Oh, you so say about vesicles. No, they were not vesicles. I think probably there are a couple papers exist about this, but I just don't remember them. So because, but it was not direct evidence that they exist in these vesicles, but they were done experiments where they managed to like mix to to different cells. One cells were expressing microRNA, specific microRNA, another cell didn't. Yet at the end, they so basically cells were labeled with GFP and RFP, right? Basically GFP1 expressed specific microRNA, RFP didn't, so they mix them, after that resort them, and they saw microRNAs which express in GFP cells in RFP cells. So basically they saw that it's possible some exchange between cells, but yeah, if I answer on your question.
Yeah, I was curious if that was true for Helium.
Oh, previous, no, I mean, we don't know, so basically. But I mean, actually, in general, germline during meiosis, it's a giant syncyty. So basically, cells connected with each other. Basically, there are like 64 cells connected, and I don't think for them it makes sense to have any other type of transfer because they share the same cytoplasm. So, yeah. All right.
Yeah, is it time to,
Ah.
no, not quite.
Not quite.
Let it go, right?
Yeah. What?
So
How
can
much
you
time
just
is that?
maybe go over the steps of the
Yeah,
RNA
yeah,
library prep?
yeah. That's what I want to do, and actually, I want to check the gel as as late as possible. So,
Sure.
because since we asked whether we can do one nucleotide resolution,
One
I just yeah.
I was just curious.
I,
We don't have to do that.
okay. Because I mean now probably it's not time for
Okay.
sure. All right. So how small RNA cloning works? I guess I'll stay here. All right, so as you saw, we started with the gel, and usually the way how we do, we run the gel, and we spike our, so this is the gel. So, and we spike our RNA samples with the radioactively labeled spikes. One spike is around 19 nucleotides, and another spike on, like, let's say, 30 nucleotides. And we run them, we visualize it on phosphoimager, or you can use like regular X-ray film. So, and since they're radioactive, right, you can visualize it, and you cut this fraction, which is 19 to, from 19 to 30 nucleotides. So basically you have 19, 20, blah, 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 up to 30, right? Everything what is there. And what we are hunting for small RNAs, which actually functional. And what they have to have 5' phosphate, right? And they have to have 2 hydroxy group. So let's say it's going to be 3', this is going to be 2'. And we can use these properties to clone small RNAs. And once we isolate this fraction, what we do T4 ligation, RNA ligase. So, and what we do first, we ligate 3' end of RNA. So basically, here we're going to have a ligation to the 3' adapter. And the way how this adapter design, maybe I'll stay here. The way how this adapter design, in fact, it's not just a regular T4 RNA ligase, it's a mutant T4 RNA ligase which cannot use ATP. Why it cannot use ATP? Because our adapter designed in a way that it's already pre-adenylated, so it has this diester phosphate bond, which kind of mimics intermediate of ligation step of RNA ligase. So basically this A is coming from ATP normally. So and since we have a mutant ligase, this mutant ligase can use only basically our 3' adapter, nothing else. So basically this ligase in fact mutant, and we do not have any ATP in reaction, just no ATP, because our main aim to make sure that only this event happened, because you can totally imagine that small RNAs can get ligated between each other like in a strain, right? But what we want to do, we just want to ligate this one and nothing else. And also what this 3' adapter has, and actually it's going to be in protocol, you'll see, because this adapter will look weird, but it looks weird because it has to be like this to increase efficiency of ligation. So, and of course at the end of these adapters you have 2 desoxy groups, so basically nothing can get ligate here. After that we run second gel and isolate only ligation products. And the way how we can follow up to follow up to ligation products because we have radioactive spikes there. So basically we can see that spikes move, so we know what size we would expect from 3' ligation. We cut the spikes and yeah, so basically they're moving like 20 nucleotides up. So, and again, we isolate from the gel and we run, and we do 5' ligation. And 5' ligation here we can already use regulative for RNA ligase because this end is totally blocked. So this is actually 3' adapter, and 5' adapter looks like, so it has just 5' hydroxy group, so just OH here and 2 OHs here. So basically it cannot ligate to itself, right? It's only can ligate to this group, so and it cannot ligate here because, I mean, we do not have anything for ligation here. So at the end, what we have our small RNA. And I mean, you have to realize that it's 1,000 different small RNAs, which is microRNAs, piRNAs, and so on. So we have small RNA, we have adapters going to be a little bit bigger. We have 5' adapter, and we have 3' adapter. And everything so far is RNA. And next step, what we have to do, I mean, we need to convert it somehow to DNA. And of course, I mean, it's only one way of doing it. It's doing reverse transcription reaction. So basically, there is RT reaction here. So you transcribe, and so this RT step, right? And at the end, you get in single-stranded DNA. And you know sequence 5', 3' adapter. So next step is just PCR. You're doing PCR, and basically you get in libraries. You still go through gel isolation, which is agarose isolation, and I mean average size of the library is 120 base pairs. And nowadays, what you can do when you're doing PCR, you can introduce barcodes during the PCR reaction. And for example, if you want to run on Salex, and we run on Salex because it's a short reads, it's not open bio where we need like long reads. So Salex gives way more reads, and I mean we care about only size 20 nucleotides. So basically, we don't really need open bio for this. So basically, you can barcode up to 10 libraries or even more. For example, HiSeq gives you like 20 million sequences. So basically, it's up to you. If you want to have a million sequence from your sample, you can do up to 200 barcodes. So that's pretty much how it works. So, and basically today, just example of one of the gel, but in general, we're going through the 3 gels. And as I mentioned at the beginning, Illumina has a protocol where they avoiding all these gel separation things. They just had 5' adapter, 3' adapter, 5' adapter, ligate everything, run only one agarose gel with PCR product. But it's really dramatically affects your library. I mean, if you just want to see your most abundant microRNAs, totally fine. But if you really want to go deep and to look for the sequences which are not that abundant, it's still better to do at least 1 or 2 steps of gel isolation. And at Cold Spring Harbor now, I would say like 50% researchers who is not working particularly on small RNAs, but they need to make small RNA libraries. They do at least first step of RNA isolation, and after that use Illumina kit. But
How many rounds of PCR do you do?
rounds PCR. So I mean, it depends on of your yield. So, but I'm trying to not go through more than 25 cycles. because PCR always brings artifacts. You always have winners. But now what we are trying to develop, and I think it's easy. I just like didn't do it yet. So we are trying, because Alexa can read like 75 nucleotides easily. What we're trying to do, you can include some random hexamers in 5' adapter, and basically what you do, you will not count how many reads you have, but you will count how many different reads you have based on, basically you will count ligation events.
All be equal proportion.
Yeah, in general, you just collapse all your reads as a single reads and treat them basically based on barcodes. So I mean, I don't know if you have random 6 nucleotides. I don't know. It's like 60 something. But I mean, you can go.
6 to the 4th.
Yeah. 6. Yeah, I cannot calculate 6 to the first. Oh yeah, yeah.
They should all be equally proportional if there is no bias. Right?
Yeah, but there is no ligation bias. It was shown. So there is always PCR bias, and basically what you can do, you can put as many random nucleotides as you want, like 6 and 7, and at the end count just ligation events rather than count PCR amplification bias, which actually still happening no matter what.
Oh, I see. So right after you ligate, you do a quick
Yeah.
sequence.
No, you still do PCR and you get in reads, and some of the your barcodes will have 100 reads for the same microRNAs, and some of them, let's say, gonna have 1. But you will still treat those 100 as a 1 because you will know that it's PCR artifact. I mean, if you imagine that you're cloning because I mean, ligation doesn't bring any bias, so.
Oh, okay. So ligation.
No, I'm just giving you example. PCR is not that bad, but I'm just giving like 1 to those 100, yeah.
So why all the gel purifying steps? Why not do just one at the end, like the Illumina protocol?
Because very often what you may get, even you have here the mutant ligase, and I mean, you're making sure that you do not get self-ligation and so on. But what you may have, sometimes you getting some, I don't know why it's happening, but I mean, enzyme's not perfect, but you getting sometimes ligation between 2 adapters. Sometimes actually they make a circular form. Whatever you have, for example, long RNA. I mean, 20 nucleotides, I'm not sure that it will manage to circularize. But
Just reducing like
you're reducing, basically complexity of the library significantly increase with all 3 steps. But in general, for people who is not working on a small RNA silencing, they're totally fine just to do first gel, and after that apply also Alexa kit, which they have. And because I mean, we have sort of in-house thing, which actually not based on Alexa, and I think it's cheaper. It's cheaper.
So if you're just running like the pre-packaged TrueC small RNA,
I would recommend to do first gel, and after that,
so even if you're getting bad reads, then all those filter out at the end.
yeah, but I mean, sometimes you, you're wasting and you getting the like most abundant microRNAs. But for example, if you're interested in low abundant microRNA, which can, so no, because I mean, you always getting winners which are highly abundant. I mean, even if you don't do any steps of selection. So nowadays these protocols work very well. So.
And you can do the gel steps non-radioactively, I assume.
Yes, what you can do, you can, I mean, now I'm running RNA markers, right? Basically what you do, you just run non-radioactive one. Basically it's going to be non-radioactive markers, right? Here is going to be your sample, which has nothing, right? And what you will do, basically you kind of cut based on how these markers run. Sure. And usually what I do, because here's the total RNA, here's pretty much like picomoles of RNA because it's RNA marker, so I still load like mock RNA, total RNA here just to make sure that it's the same mobility. Yep. So, but Yeah, you can do that, but we use radioactive one. And I mean, no one asked me actually what happens with these RNA spikes because they're pretty abundant. So, and I didn't tell you up in front. So basically, both of these spikes have a restriction enzyme for very rare endonuclease, and we do an endonuclease digestion after PCR step. Bless you. So it means that we may lose some microRNAs or piRNAs which have this nuclease signal.
Six-base pair inhibition sequence?
Either 6 or 8, I don't remember. It's a PME1, I don't remember. Yeah. We don't lose a lot, but if you're particularly interested in microRNA which has this site, yeah, it's going to be a little bit sad.
Quality of it, when you look at it, like if you assess the quality on a bioanalyzer, I guess, do you do it that way, or how do you
Assess the quality. Basically, if you have any RNA degradation in your sample, you will see that your ribosomal RNA is degraded. So you run bioanalyzer and you see it. So basically, if ribosomal RNA is degraded, most likely small RNA is
So you
degraded.
run total RNA before
You can run. At the beginning, I mean, 3 or 4 years ago, we didn't have bioanalyzer in the lab, and we didn't use it, and we were fine. So, but
just run it just like kind of denatured gel, just like you would total RNA?
yeah, you do the natural gel and so on, but I mean, actually, our lab didn't have a problems to waste, let's say, $1,000 on degraded samples. But yeah, if you really worry about this, yeah, I would recommend to run the natural gel or even polyacrylamide gel like this and see.
But I guess I'm just saying that there is a QC step before you, I mean, if you're going to be sequencing.
Yes, there is QC step.
You're basing off of the ribosomal RNA.
Yeah, yes.
That if it's intact.
Yeah, if ribosomal RNA is intact, most likely your small RNA is intact because actually for bioanalyzer, right, for majority small RNAs, you will not see them on bioanalyzer. You have to run like either nano chip or something like, or high sensitivity chip, which actually, I mean, they're expensive, so it's like, and not necessary you will see something. For pachytene pyranases, I know you'll see them, so, but I mean, in majority cases, I do not see on a bioanalyzer peak of small RNAs because they're not that abundant as mRNA or ribosomal RNA. So, I mean, yeah. Any other questions? Yeah, I mean, we can try to see. So if we do not see nice separation yet, because I know basically bromophenol runs approximately 10 nucleotides, xylensanol it's 30 nucleotides on this.
Propanol is the faster one.
It's the darker blue, right?
The darker one, right?
Yeah, basically we already have small RNAs in the gel, so we can try to see them. Of course, it will not be nice separation from tRNAs. Okay.
Wait a little bit.
Yeah, let's wait. So
Or-
I have something to show if you
Yeah,
guys
sure.
kind of finish grilling him. So let's thank
Yeah,
him for
yeah.
that fantastic presentation.
Yeah.
I'm gonna put on something on my computer. Is that okay? I just want to show 2 slides.
Oh. Where did I put it? Oh, in the pocket.
Let me just show you something. Something. Suspense. Is the bar open yet? Oh well.
Oh, that's why it's 5 o'clock. I thought why it's 5 o'clock so
Can
precise.
somebody help me put to the projector because I'm totally stupid when it comes to this stuff. Oh, I need my backpack. That I have.
Oh, I see.
Thank you. Thank you. Yeah, after picking up this, Sue and Chunma and I had organised that Chunma and Blake and I would get everything up to Went, but now Chunma's not here either.
Now what happens? Oh. stop miking me. I don't want to be mic'd. Please. Please.
So
How do you flip it?
which one is that? That one's the computer, I
I don't know, because that one's still showing.
guess? Yeah, this one's
Oh, there it is.
Okay, there you go.
All right.
This is
Another
your
high-
high-tech device.
So, this is a high-tech device to hide the data.
Yeah.
Because you're dying to see it.
Oh.
Oh! How much do I get?
The PCR.
Gin and tonic, margaritas, fine.
Whatever you want. Name your price.
One of both, one of each. So Eric ran the libraries for us, okay? And, okay, don't get nervous, okay? Even though there's not much here, there actually is enough.
Okay.
Okay, so you can't see it on the bioanalysis trace, right? But if I show you the peaks, there is enough there, he told me. The only one, this is raised, there may not be quite enough in that one. But they have tricks. And he said there probably is enough to get data. So you can see the range of sizes is exactly where we want it to be for the ones that are very, very bright. There is definitely enough here for Judy's also, he told me, okay? And that's the first 12. This is very exciting. After all that craziness, I'm just shocked that there's anything there. And all of them work Agnes. Even the ones, so originally Agnes's samples, they'll send back, they're like, we're not running these. There's no RNA on the bioanalyzer from the RNA starting step. And she pulls an article right when I'm trying to prepare the Excel sheet for them to make the libraries. And she said, Mona, read this. And it said that insect RNAs may not run right on the bioanalyzer chip, right? And I said, Eric, put them on. And so libraries were prepared. You can't quite see them. It's this, but they're here actually.
It's there, really?
Oh yeah, they're there. The peaks are there. They're totally there. Okay. So everybody has libraries made from the ones that were done yesterday. So whatever was there is going to get data. Okay. It's amazing. I am shocked. Really, because there was a lot of stuff going on over there that you were watching like, gloves,
So
what's
these
going on?
are the ones you did yesterday?
Yeah,
Oh, okay.
2 days.
Right.
These are them.
So
That's why I'm so surprised.
Ask one first, right?
Yeah.
So,
Let-
just to know, why is my sulfane?
I'm going to let me show you.
The input RNA?
Let me show you, because if you look at these guys, if you look, I couldn't capture this. I tried, but it was hard. So if you look at these, if you look here, let me unclick.
I can see the bump.
You can see the bump. That's all you need to see is a little tiny bump, and it's going to be fine. There's bumps. That's all that matters. He knows what to do. Don't worry. There's big bumps in most of them, right? But
Because you don't know, remember that they've got to quantify the exact amount.
exactly.
Yeah,
So
those
now,
ones probably have a ton extra.
yes, a ton. So now they first do qPCR. Remember, so they can get the exact amount of DNA to load to make the right cluster densities. So after the qPCR, he'll have a really good idea of exactly how much DNA there is. And that's going on today and tomorrow.
Like P5 and P7?
I think so.
It can't be organism dependent.
No, it's not. It's definitely not. It's
Or
the
whatever,
two.
something
It's the
in
two
the
adapters. Exactly. Yeah. Yeah.
So
And
I still need to know what to do better so that I have a better distribution.
yeah, we'll have to talk about that. Yeah.
So can you go back?
Because
couple-
yeah, of course.
Well, finish your thought.
No. I was just saying that.
And that might be useful to other
Well,
people.
we didn't look at your. I mean, you have to figure out with insect RNA what a good way is to assess the quality of it, the total RNA.
Yeah, I think I was going to send an email to the insect base.
Because I don't know. Okay.
Yeah, and find out.
I mean, people make Drosophila RNA over time. You'd think there's got to be standard protocols, right?
Oh,
And for checking it.
I don't think my protocol is bad.
No, I don't think so either. But we need to check it. If we can't run it on the RNA chip on a bioanalyzer to see what it looks like, you want to know the integrity before you start. That's all I'm saying, right?
No, I think.
Yeah. Yeah. You want me to go back to the PowerPoint?
Just go back 2 slides.
So that's the actual PDF. Let me go back.
Go back to the previous.
Yeah.
So can you just explain to me what it is I'm actually looking at? So if you take like my 4.
Yeah.
2 of them have darker bands, one's higher, one's lower, 2 of them have slightly more
Right.
diffuse bands.
Exactly.
What information
Because
can
so
you
the
get from
RNA was fragmented chemically. Okay. Chemically. So it breaks up into different size
Right,
fragments.
One of the first steps,
Yes.
The
Yeah.
Michael did,
And
right.
then, depending on the exact input of amount of RNA, how perfectly can you really quantify it, right? It's not exact, but if you look across, like, they're pretty close. They started with a certain number of nanograms, right? And then the amplification steps that occur during the PCR cycles, some more or less, and some fragments bigger and smaller will amplify better, right? And so you're going to get a smear of amplified PCR, and you'll have a little more length.
What's the range, like 250 to what?
250 to 450 is what they usually look for. So those are a little bit big, but the peak is really nice right in here around 350, so we're good. These are beautiful. They're really good.
Okay, so this ties back actually to the question that I asked, I think on Monday, about if the fragments are random, how do you know that they're this size? And if
Yeah,
you
oh
have
no,
to do
yeah,
that-
this is the
so
way.
this is kind of the answer
Yes.
to that. So
Yeah.
they're not, this is the range of sizes, and
Yes.
the ones that are more diffuse just have a bigger range.
Exactly,
Got it.
exactly. So we did 12 cycles of PCR. The protocol calls for 15, but I used to do only 10, but he said do 12, so we did 12.
Okay.
But it's still, that will have reduced in PCR duplicates, so that gives much less noise in the end and better data, because you get rid of the PCR duplicates, you don't want them in there, right? So this is perfect. So this is looking okay.
And so if you get a
Uh-oh,
sharper
nothing.
peak or a more diffuse peak, is it better data one
No.
way or the other?
No,
It's just
it's
the
really
range.
not.
It doesn't matter.
No, it doesn't
Because
matter.
all that matters actually is that end that's binding, that starts your cluster.
Exactly.
So it doesn't really matter
Ex-
how long it is.
Exactly, it doesn't matter.
Okay.
It doesn't.
Okay.
Does the bioanalyzer, like the peaks that you see, is it reading absorbance or what is it?
Yeah, it's reading fluorescence intensity, so it's mixed with some kind of fluorescence
Okay.
thingy. Yeah,
Okay.
fluorescence thingy. Scientific.
Dye.
Yeah, there you go.
Is it cybergreen or something like that?
I don't remember what's in the mix.
Oh, okay.
Proprietary, as usual.
So there's still hope for my question mark sample?
Yes, there definitely is.
Okay.
He told me, I'll show you the email. He said there's definitely hope.
Great.
The qPCR will tell us more.
Yeah, okay.
So if we haven't already got data,
Great.
we're not on that track, we're not getting data.
Well, there
Right.
was there was an earlier run.
Yeah, because
I don't have any data on
you don't have data up on your iPlant yet. Do you know about that?
Did you look at it, it's gotta be the NGSCs. You can see if it's there, you can't
So-
necessarily transfer it over.
Yeah, so
After
from the
tech you're working through.
From the, it should. So wait, I know why.
But if you saw your name on that email with the pass filter that one
Yeah.
you got before,
Yeah, wait, did you send me a second batch of RNA? No, then yours are good, you're there.
But they can
They may not be up yet because it takes time to get them there,
Yeah,
that's
before
all.
before we leave this room, I just wanna, when you guys are at that last few moments, I wanna
Yeah.
just go through again.
Yeah, so they're not up yet, that's all, but they were done and they were good. Okay. So, yeah, don't worry, no, there's data
A
there.
little bit of confusion.
No.
In the second group, because
I'm
there
sorry.
was a second group of the first
That's
shipment,
true.
and
That's
the
true, I know.
So all of the first shipments work?
All of the, no, I don't wanna say, no, not everybody's RNA from
Okay.
the first shipment were good,
I haven't
so
heard
those didn't
anything
go
about,
on.
I didn't hear anything about my RNAs from the first batch.
We ran 2 by 2 for you, and they were fine.
They were fine, okay.
Yeah, of course. Yes, that's
Okay.
right. That's true. I guess I should have sent it, okay, you're okay, I didn't do that. I only said, you didn't do that good. But it's okay, we're
Okay.
learning. No, I'm sorry. And so you wanted to know about your next shipment though, didn't you, Ray? I actually have the data right here and I
No,
can show you.
that's it, the stuff you just showed, that's my second shipment.
Yeah,
Yeah, so that's,
but I
the
have,
first 2 by 2 is the most important to me. So
Okay.
that's okay.
And there's
The
data
second
there.
one was the brain and cornea, is that right?
Oh,
Is
that's right, I
that
did
what
have a,
these- Is that what these four that you're showing now are?
no, so there's
I'll,
they're actually, there's no brand, none of the brands went forward. They didn't pass
so, yeah,
the initial
I
bio.
did tell you this, Ray, right? The original ones you sent were here,
Yeah,
and a couple were not good.
yeah, and then I
And then-
resent a bunch in
Yeah,
the second
and
batch.
some of those were not good either, apparently.
Yeah.
But that's all right.
That's fine. No,
So then I
okay.
wrote prepare in the column where those were that were made, you see?
Yep, yep,
These
yep.
were made first, and then these were made
Perfect,
just,
yeah, that's what I
here.
thought, okay.
okay?
I just
Anybody
kinda-
else have
I
questions
lost
about
track of
this?
that whole thing.
You know, carriers are good.
I haven't seen it on there.
Just double check.
They did the last 2 days that she just showed us.
They were 2 additional recordings.
Yeah, yours are all done. Yeah, they're all done.
Yeah, I knew that there was an issue.
Yeah, yeah.
One of them was,
So, all right?
Mine were,
Yay.
mine were okay, I think, because
Yeah.
I didn't hear anything, so.
according to my
Give me a sec.
nano chief knockoff of the nano drop.
Because I saw some of mine up there, yeah, okay, great.
So
Okay.
I think that's the
Yeah. Oh, so you didn't see your traces?
Some of mine, well, no, some of mine, no, I saw my traces.
You saw your traces?
Yeah, yeah,
Oh, okay. Yeah, yeah, yeah.
There's no
No,
bands, but the traces, little bumps.
the traces are good.
Yeah.
Vasily
what I
Vagin,
really wanted was 2
that one library of mine that didn't work very well on Friday,
It's being rerun.
so it's been run now, it will be run Friday. I mean, I just need to know if I should be looking for it or no, not yet.
Yeah.
Okay, that's okay, that's the only
He
question.
said to me, I know, he said, I'm not sure when I could put it on. All right. So I don't think it went yet. He'll let me know though.
It's okay, this is
Sorry,
fine.
yeah.
Doesn't matter.
All right, cool. Yay. We're done with this? So what do you, oh, the gel's still running? Oh, 5 more minutes, not too bad. All right,
you know,
cool. Okay, all right. So, are we okay with this part? Like, any other questions? So I think part of the thing is to like discuss how to really get good RNA samples, all different protocols that are available, how to get rid of DNA, and that kind of stuff, so that we could really have protocols for the students also, right? To just have an idea what it takes to do that. So how do you think we should do that? Like, if each one of you write up a really great protocol of how you made RNA, would that help? And then we share those? What do you think? And especially the tips, right? That's the most important thing, like tips. Like, yeah, you can't look at this funny because the RNA will degrade, right? Those important
Literally,
things.
our lab, we found we can't do more than 4 at once.
There you go.
I mean, so,
This
this
is-
is very
yeah.
simple, and
Yeah.
they cannot do more than 4
I
at once.
believe
And also,
that.
never drop their sample size down, some in the morning, some in the afternoon, quality went flying.
Yeah,
And I mean,
it's
who
amazing.
would, you know, I mean, that's.
I don't know why that is, but
I don't know why,
yeah.
but I can tell you. it's-
And the other thing that I found is that if I make a lot of RNA when I'm on a good day and I get like micrograms of it, if I freeze that one tube I always freeze RNA -80, never -20, never. And if I don't alipod it and I freeze thaw that RNA tube more than 3 times, it's gone. Gone. So I alipod it -80 into 72, so it's a pain, but RNA's not easy. Why we should work on DNA.
let's change over to DNA methylation.
DNA,
sequence.
there you go.
I think that'll be
DNA,
fun.
so you know, dinosaurs, we could do DNA, it's crazy. So, so there's little tips about, I don't know what else we should include. Bruce, you have a thought? No, you were about to.
I was gonna ask you your recommendation to de novo sequencing of novel genes.
Oh my goodness, yeah, we could do that on the Pac Bio, right? Yeah.
So you want those, upload those to the
You upload, you want me, you want me to upload
No,
the,
I'm saying RNA prep protocols,
yeah.
do we upload those to the?
I think that's the best way, right? You guys are comfortable with using the wiki and everything now, or what do you think? Yeah? It's half and half, I know. But then again, you know, if you buy the Qiagen RNA Easy Kit, the protocol is so well written out. It's just like, just follow this, and that's it, you know? Right, Zymo is
We
easy,
actually make our own trizol.
and
it's
I
so
do
much
trizol
cheaper,
only,
and,
I know.
you know,
And the trizol protocol is so simple, just follow it, right? You don't think so?
We made a couple copies.
Oh, so then maybe those would be good. Oh, and actually, the small RNAs, right, there's a special trizol, isn't there? Oh, Young used to use, you know, Young Park used to use a special trizol. Oh, you do kit also. But kits are always a lot of money for people. Yeah.
The ratio.
Yeah, so it depends. It's but if you're not gonna focus on small RNAs right now, but still, I find trizol is the best. I just do.
But
right? Oh my God, I
So
know.
is he more of like a, this is how it works?
He's a
kind of
machine guy.
Yeah,
But I didn't know this coming in. I really didn't. And I specifically asked Eric, who should know, and him. I said, "You sure you can do this?" He said, "Absolutely." Because I wanted Eric's technician to do it, who prepared the first libraries. He said, "No, Michael knows more than Prashant.
Yeah.
There's no
It's
way."
always when you watch them looking at the manual so
Yeah,
much.
exactly.
Well, we're glad
And then he mentioned Eco R1 for one of the polymerases. Did you hear that?
Yeah, I heard that.
You did?
I kind of ignored it.
I was cracking up. Did you notice that he said that? Somebody asked him about the polymerase. He goes, "You mean Eco R1? He meant the whole life. But he clearly he's not a molecular biologist anyway. But it worked, so we're good. Exciting. I'm really excited. And you were so nervous about your RNA. Oh.
I've never done this before.
And look at you. You're pro. Pro.
What did you get to do? Your phone?
Yeah, my graduate students were off the conference. So I couldn't
That's
even
really
ask
great.
where things were in the lab.
Really great.
You know, where they stored stuff. You know, which freezer did you put it in?
All right, cool.
I have a number of colleagues that have done RNAs.
He's doing his next step.
So I was asking them, but there's, it's all really different. So one of them does plants, cuts the leaf off, sticks it in the ground, and sends the entire thing out again and do it again. She's never done anything molecular in her life until she started her analysis. That's very tight.
Yeah, there's different flavors.
One of them was I went to study with turtle, and he's doing turtle heart and turtle heart and brain, but they're big, you know, so he's got a whole heart, so he practices up in a mortar and pestle and gets blood.
But I think that,
You know, and I do have, then it occurred to me, I knew someone over at Washington, I do it for that. Super busy.
well, and that's the thing, any small RNA does.
Shemang Chen.
Oh,
Yeah.
She's always busy.
Yeah, she's always really busy, so I really hate to stuff that.
I don't know.
I feel like my
Yeah,
students
I
don't know.
got
You know, kind of thing. I mean, yeah, but you know, so right, so there was no doubt that I was gonna be prepared. But it was things like, so how do I homogenize it? So
Yes.
I understand if you do too much, you get sharing, and that can be really bad.
Okay.
So,
Exactly.
she's got,
Yes, and it's easy.
So I guess, are we waiting for doing for
Stay yourself for 5 minutes and then we're gonna head over to McCook.
did,
Do you have the bee stain?
mixture and sonicated, sonicated.
So I don't know if that's not quite enough time to make this one.
So we did a mix, we vortex for 30 seconds and sonicated.
No, we only have one bit. I thought we might be separate.
That's good. I mean, honestly, it's probably overkill. I avoid that. What we used to use was a little juice polisher, like one of those little blue mini tools. And you can use
Yeah.
that. You really don't need much.
Alright, so now what I'm gonna do, so I'm just gonna put the cyber ball
Okay.
into TB buffer.
Can you say that again? I'm sorry.
Okay. Okay. Yeah, any it's sort of fine. So, ready?
Yes.
All right.
Camera,
So-
action.
Yeah. All right. So we're gonna put cyber gold into TB buffer and stain our gel for 5 minutes. So first I will put cyber gold. It's light sensitive, so that's why I have aluminum foil. And I put in a little bit higher than we needed, like 100x higher, just to make it.
A little bit. It's all relative.
Yeah, everything is
That
relative.
looks like rubidium, that color is
Yeah,
interesting.
but it's not. I mean, in theory, we could try to use ethidium bromide, but it's less sensitive.
Yeah, this is more sensitive. Cool.
All right. For now. All right. So now gel disassembly. Do you have sink here? Yeah, I have. So Yeah, I have to get rid of TB. So. Yeah, and usually I have a nice gel separator. I don't have it now. I'm gonna use a razor blade, which actually I don't recommend students to use because they crack the. All right. And yeah.
Yeah, exactly. He's pro at this stuff. I used to do the long sequencing gels with P32.
Yeah.
2 millimeters, remember? Oh, those were terrible.
All right.
You know, when we moved,
with the chair in the middle.
Ah, the worst.
when we moved, I actually found that I had a stack of those
Really?
snakes
Yeah.
still in my lab.
It's crazy.
All right, and I will cut a little bit gel because we don't need that much.
Okay.
All right. And since it's 15%, so it's pretty. Since it's 15% gel, you can be not gentle with it. So it's very difficult to break it, but don't try. 15.
I'm sorry?
15, 1, 5.
Oh, 15.
Yeah.
You can understand.
Yeah. But be careful, sometimes you can slide on it, so it's.
Oh, yeah. We have undergraduates. I'm sure they can share it.
All right, so. Yeah, and normally we're using shaker, but today it's going to be manual shaker, so it's like.